Hello students, in the previous part 5 video, we have seen Mendel's first law, that is law of dominance. In that law, we have seen when homozygous pure for their genotypes are crossed, parents are crossed, then in the first generation, all the offsprings expressed only the dominant characteristic feature. Now, what happened in the F2 generation? Mendel has observed appearance of some of the phenotypic characters that are related to dominant parent in the F2 generation. Then it was concluded that though the factors of the alleles that are expressed in the F1 generation, that is because of not mixing up of the alleles or the factors, but those factors or the alleles just they stood together and they got segregated or separated in the F2 generation. That we will write in the form of a sentence here. In F2 generation, Mendel has observed appearance of dominant phenotype or character as if that of F1 generation, and also recessive character which was not visible in the F1 generation, which was not visible in F1 generation. Then we can conclude that alleles did not get mixed up. Then it was concluded some factors which are responsible for inheritance, did not mix up, but they all stood up together. And got segregated in F2 generation. Now, this total concept was explained with Mendel's second law, that is law of segregation or Mendel's second law. Mendel's second law. How can we see this one in the form of a definition? So, the allelic pair of a hybrid that is segregated or separated during the formation of gamete, and that during formation of new gamete, it received only one of the two factors, and those two factors are pure for their genetic trait. Now, see the definition clearly. The allelic pair in a hybrid gamete, in a hybrid segregated or separated during gamete formation, and those new gametes received only one of the two factors of a pair and are considered as pure for a genetic trait. This second law is also called as law of purity of gametes. This law is also called as means Mendel's second law or law of segregation, also called as law of purity of gametes. Law of purity of gametes. Now, what example can we take this to explain this Mendel's second law? So, example taken is again monohybrid cross. Monohybrid cross. Here we can see the best example. Likewise, in the first thing, what happened? We have taken the parents which are nothing but homozygous dominant crossed with homozygous recessive parent. Now, we also have seen the obtaining of parental gametes, like gamete has obtained only one allele. That only we have written. The new gametes received only one of the two factors of a pair. This is a pair of alleles, but in the gamete we have got only one allele or one factor, like this. This we can consider as a best example to explain the definition. We have to see what happened in the F2 generation, because in the first law we have seen only up to F1 generation, means first filial generation. Now, if you see again the cross, F1 generation, we have got all the offsprings as heterozygous. Okay. So here all are of genotypically heterozygous, and phenotypically all are expressing the dominant allele and the recessive allele. Okay. Now, when we self-cross F1 heterozygous offsprings, F1 heterozygous offsprings, we used to get, so means the heterozygous offspring which is obtained in the F1 generation is self-crossed with each other. Now, when we arrange this one in the form of a Punnett square, this one we also, according to Mendel's second law, we also said that gametes got separated. So like this, both the heterozygous alleles got separated into separate gametes. Like this. Let's draw the F2 generation Punnett square. Let's represent the dominant and recessive alleles in the Punnett square or checkerboard. So here, instead of getting all tall plants, here in F2 generation, Mendel has observed the recessive one also. So in F2 generation, the recessive character or phenotype also appeared as if parent, as if recessive parent. As if recessive parent. So by this, he got some conclusion that some factors or elements, according to Mendel's language regarding the genes, some factors or alleles which are responsible for so and so phenotypic character got separated in the F2 generation, and then they are expressed. Now, because in law of dominance and law of segregation, we have seen only one type of character, and we have taken only one type of character for our explanation, and we started doing that. But when two different characters are seen in the offsprings got transmitted from the parents, because in Mendel's time, Mendel did not do any lab experiments, and he don't know about the genotypes, but Mendel gave everything, the concept and the contributions, based on the phenotypic characters only. That we should remember. Third one, law of independent assortment. This is considered as the third law, and this law is applicable for the genes which are on different chromosomes. This sentence is considered as an MCQ. This law is applicable for genes or alleles on different chromosomes. Different chromosomes. This one you can remember as an MCQ. Which law is applicable for genes on different chromosomes according to inheritance? We can answer Mendel's third law, that is law of independent assortment. Now, this law is based on dash. Again, another MCQ we can consider. This is based on dihybrid cross. Dihybrid cross. Now, what is meant by dihybrid cross? See, when we are considering only one type of character, it can be one of any of the 7, either height or flower position, etc. and etc. But when we are talking by mixing up of 2 different characters, like it can be of height of the plant or color of the flower, or it can be height of the plant or shape of the seed, or it can be height of the plant or with that of color of the seed, and it can be height of the plant with position of the flower, etc. and etc. So we can mix up any 2 characteristic features or phenotypes, and we can explain this law of independent assortment. So we'll see what is dihybrid cross. So dihybrid cross is a cross between 2 pure parents that are differing in 2 pairs of contrasting characters Now, this one we can say as one type of definition, because I told you there are different modes of explanations and definitions for explaining these laws. First one, in the first law of dominance, I gave you 2 types of definitions also. Now, we also see third law, or this law of independent assortment states that the allele of 2 different genes got sorted into gametes independent of one another. Now, third law states that the alleles of 2 different parents, we can write, or gene forms got sorted into gametes independent of each other. This one is also considered as second type of explanation for law of independent assortment. Now, another type of definition is also there, that also we will see now. When a dihybrid form of gametes with assortment of alleles, means the gametes containing different types of alleles, that alleles are of different traits, in the sense that are having different types of genotypes and are expressing different types of phenotypes also. That gamete is independent of the original combination of the parents. So we will see one of the best example under dihybrid cross, which is given according to NCERT, that is shape of the seed versus color of the seed. Now, shape of the seed is given with the genotypes that we have given, but Mendel did not give this genotypic ratio. But we are, for our convenience, we are explaining by taking the allele forms. There are these 2 pairs. There are 2 pairs of alleles. One pair of allele is responsible for dominance. So the dominant phenotypic character of the shape of seed, right, okay. Now, dominant phenotypic trait is around the shape of the seed. Dominant trait or character or the phenotype is round shaped seed. This pair is the genotype of wrinkled shape of the seed. Now, color of the seed. This pair of alleles are responsible for dominant feature dominant feature or trait with yellow color of the seed. Yellow color, which is considered as a dominant color for the seed. Now this small y representation or the allele pairs are responsible for green color of the seed. We are taking two different characteristic features for our explanation. Now, let's take two types of these phenotypic characters as parents and do the cross. So these are considered as the parents. Now, let's separate or sort out the gamete forms of these. Now we are writing here parental gametes. Parental gametes. How did I take this one? For suppose, if the arrangement of alleles are like this on one chromosome, means which are one is responsible for dominant and recessive alleles like this. I am crossing this one like capital R, small r. Again another one, capital R, small r. Capital R, small r. Means dominant allele, recessive allele. These heterozygous form of allele distribution only I have written here. Here also we can do the same. Like this. So we have got here one type of gamete or allele distribution into the gametes. Here also one type. But the total number of gametes overall when obtained are 8 in number. But here the types of gametes are only 2. That I will write here. Types of gametes when taken or when compared to parents are 2 in number. Total number of gametes obtained. Number of gametes obtained are 8. When we try to obtain the F1 generation, we have to combine or fuse these two different type of gametes from two different parents. Now, if I write this in the form of a Punnett square, how does this look like? We have to write, first of all I will draw the board. One gamete, another gamete. So here we are getting both dominant allele forms. They stood together. Okay. Means they are getting expressed independent of each other here. In a sense, R is dominant for its shape of the seed. Y is dominant for its color of the seed. But both are obtained in the same offspring. Means the plant which is showing here the type of phenotypic character are, the seeds in that plant are showing round color of the seed and the yellow color of the seed. Both are dominant characteristic features. So here we are seeing all same type of alleles. We can write answer for this as all obtained F1 offsprings are expressing are expressing a round shape of seed. This one we can also write like this. Any expression is correct only. And yellow color of seed. Since we know that capital R, which I have written here, is nothing but the expression of the dominant allele. Small r is expression of the recessive allele. And dominant means which is suppressing the other type of its contrasting genotype. And recessive means the one which is getting suppressed in presence of its another contrasting allele form. So here, if we fill also, we will get the same thing. If we come to the phenotypic ratio of F1 generation of dihybrid cross, we have to represent like dominant is to recessive. Since all offsprings in F1 generation are showing expression of a round shaped seed with yellow color, we are writing 16 number of offsprings that are obtained here. No recessive means expression of the alleles is seen. So the ratio will be 16:0. Coming to genotypic expression, the ratio will be all are having one dominant allele of the shape of the seed, one recessive allele of the shape of the seed, another dominant allele for the color of the seed, another dominant allele of the color of the seed. So here all we got capital R, small r, this one, capital Y, small y only. We get these are 16 in number, and we did not get any of the parent type like this. Capital R, small r, capital Y, small y. That type of genetic expression we did not see here in F1 generation. We know that for obtaining F2 generation, we have to self-cross all the F1 offsprings. Things are self-crossed. So that one we can write as let's sort out the gametes from this. So F1 generation gametes. This one we can write as capital R, capital Y in one gamete. Now, capital R, small y in another gamete. Small r, capital Y in one gamete. Small r, small y in another gamete. Now, here also the same thing. So the gametic distribution is like this here. Since we don't have sufficient place for this one, I'm erasing these gametic forms and I'm writing directly the Punnett square. Here we can write one best example, an MCQ like. Okay, so this is the expression of the gametes. So here if you write one an MCQ, the types of gametes from heterozygous F1 generation, types of gametes are how many types? 1, 2, 3, 4. 4 different types we have seen are 4. This one you can learn as an MCQ. Now, number of gametes that are obtained. Number of gametes that are obtained are 1, 2, 3, 4, 5, 6, 7, 8. So 8 number of gametes. These both you can learn as an MCQ. We have seen just before the different types of expression of the gametes that I am directly writing in Punnett square to explain the F2 generation. So F2, these both are crossed. So we have obtained different types of gametes. One is capital R, capital Y, capital R, small y, recessive allele, dominant allele, again recessive allele, recessive allele. Here also like dominant allele, dominant allele, dominant allele, recessive allele, recessive allele, dominant allele, recessive allele, recessive allele. See, though both are considered as the dominant character expressing alleles or the allele pairs, both are seen in same plant. It means that the dominant alleles, though they are together in a same plant, they are expressing themselves independently according to the definition of the dihybrid cross. We can write this one. This is the condition about the different types of genotypes that are obtained in the F2 generation. Now we will sort out some MCQs from this distribution of gametes and also obtaining of the offsprings. Now, for suppose, if we ask a question, total number of offsprings, the answer will be 16 according to this distribution and obtaining of the gametes. Now, how many of the offsprings are expressing their genotypic character or genotypic allele distribution similarly exactly like that of parents? Genotypic exactly like that of parents, not F1 parents. The first parents which we have taken just before the F1 generation. Those are only 2 in number. That also we will write. The number of offsprings that are having genotypes exactly like that of original parents before F1 generation. Before F1 generation. Now the answer. Will be 2. What are those 2? This one. Capital R, capital R, capital Y, capital Y. 2 dominant different pairs of alleles. Now this one also. This is also another type which are expressing the parents. Not the F1 generation, because here some doubts will be coming to the students. Madam, we have taken this one as parents. No, we did the F2 generation. So I am asking you the question very clearly. The number of offsprings having, because these are the offsprings that are obtained by self-crossing of F1. Okay, we can consider now as parents. But clearly I have represented here original parents before F1 generation. So the answer will be 2. What are those exactly? These 2 are expressing like that of the first type of parental type, which are having this type of genotype expression. So those number is 2. Now let's write another MCQ. How many of them are phenotypically expressing round shape of the seed with yellow color of the flower? That we'll see. Round shaped with yellow color of the seed. How many are there? So this one, 1, 2, because here there is one dominant allele of both the different types of phenotypic characters. 3, 4, 5, 6, 7, 8, 9. That number is 9. Now, how many of them are expressing? According to the gametes, first of all, we'll see. Round with a green color seed. Round shape with green color seed. How many are there? Round shape with green color. So 1, 2, 3, 4, 5, 6, 7, 8, 9. This one we have seen just before. Now, round shaped with green color. We can write 1, 2, 3. So their number is 3. They are not like that of parent. Parent is having phenotypically round shaped yellow color expression. Now, another one. Here is the wrinkled shape of the seed with a yellow color. 1, 2, 3. So, another phenotypic character. A wrinkled shape of seed with yellow color. With a yellow color. How many are there? The number. So 1, 2, 3. So that is 3 in number. Now, if we see the last one, wrinkled shape of the seed with a green color. Fourth one, wrinkled shape with green color. Answer is only 1. So here round shape with yellow color 9, round shape with green color 3, wrinkled shape with yellow color 3, wrinkled shape of the seed with green color 1 only. From these numbers, we can easily frame out the phenotypic ratio of F2 generation of dihybrid cross. Phenotypic ratio is how many? Round, yellow, 9. Just I have given explanation about those. Round and yellow, etc. Next, round with a green color seed, 3. Again, wrinkled with a yellow color, 3. Next, wrinkled with green color, 1. So the phenotypic ratio of F2 generation of dihybrid cross is 9:3:3:1. Now, coming to genotypic ratio. Here we can write this type of allele is only 1. So 1:. This type of alleles, capital R with capital Y. How many are there? 1 and 2. So we can write this one. 2:. So how many of such type of alleles are there? Capital Y. Okay. Capital R, small r, capital Y, small y. How many are there? 1. Again 2. Again 2 only. This one. See, this one and this one. I am writing as again 2. Now, capital R, this one. 1, 2, 3, 4. So that is 4. Now, so we have finished this, and this. This also we finished. This also we finished. This also. This also. How many are left over? The remaining one. Capital R, small y, small y. No. So only one type of capital R, small y, small y. This also we have finished. Now the genotypes, allele pairs, how they are? Capital R, small y, small y. So again these are 2 in number. So this also we have finished. Now coming to wrinkled shape. How many are small r, capital Y. Again 1. So this we have finished. Small r, capital Y, small y. How many? Again 2 number. Now the last one is 1. With wrinkled shape means small r, small y. That is 1. So we can frame out this one. Here I have written as 1:2:2:4:1:2:1:2:1. This one we can easily frame out like this one we can bring here. I can flip 1 and is to 2. This one also I can flip like this. 2:4. Just for our convenience to remember. Likewise, we can now explain this one as 1:2:1, 2:4:2, 1:2:1. I did not change any of the numbers in the ratio. Just I have flipped for our remembering means expression. Okay, 1:2:1, 2:4:2, again 1:2:1. Like this we can easily remember. Now we will see another type of allele forms. How they are asked as MCQs now. In this, the parental phenotypes round and yellow are 9 in number. Now I will tell some of the MCQ models here, which there is a possibility to ask questions from. Okay, now let's take round shaped with yellow color of the seed are 9 we have written here. So 9 are of a parental phenotypically. Then what about this? Round is to green color of the seed. These are considered as the recombinants. Recombinants. Another one also. Wrinkled shape with yellow color of the seed. These are also considered recombinants. How many are recombinants? 3:3. Totally 6. Totally 6 offsprings out of the 16 are considered as the recombinants. Now, if a question was asked like, what is the percentages of different types according to the phenotype 9:3:3:1? That we will write here. So, the recombinants. So, coming to percentages of phenotype ratio, that is 9:3:3:1. We know that total offsprings are 16. Offsprings are 16. So if we do a little bit mathematical calculation here, since 16 is considered as the 100%, what about the 9 number of offspring? What will be the percentage here? If we do cross multiplication like this, we can express this one as 9 into 100 divided by 16. We will get an answer if you do calculation 56.25% of offsprings out of the total 16 are considered to be having round shape of the seed with the yellow color of the seed or cotyledon. In the same way, if you do calculation, since 16 is considered as the 100%, what about the recombinant first 3 round shaped with the green color seed? What will be the percentage of the 3 number of recombinants? So here again the question mark. Answer will be, if you do calculation, you do later, but I am writing directly the answers here by doing this small type of calculation which I have shown for the 9 number of round and yellow shaped one. This also will be of 18.75%. Now, the last one, that is a wrinkled shape with a green color seed. What is the percentage? If you do calculation like this, 16 if we consider as 100%, what is the ratio of one number of the offspring that is showing either dominant or either recessive one? Purely. So this is about the percentages you can learn for your MCQs. Now, we can frame out one more concept regarding this dihybrid ratio, that is, new combination in F2 proves the law of independent assortment because all the though the dominant alleles are side by side, they are getting expressed independently. Next, at this law, as this law is applicable only for the traits on different chromosomes, this law is not universally applicable because we have seen some types of expressions in the F2 generation, means when we are self-crossing the F2, basing on the F1 offsprings. We cannot say the crossing happens only between the chromosomes which are alleles which are on same chromosome, alleles which are on different chromosomes. So now that I will give one more sentence. Please learn these sentences for your MCQ statements also. That will be very much useful. Now, new combinations of F2 generation proves the law of independent assortment. Now, this can be this can be a good statement to learn for your MCQ point. As this law is applicable for alleles or genes on different chromosomes only, it is not universally applicable. It is not universally applicable. We have seen law of dominance also not universally applicable, but law of segregation is universally applicable. Now we have seen 3 different laws. In those 3 different laws, law 1 and law 3 are not universally applicable, but second law, that is law of segregation, is universally applicable law. Next, about a tri-hybrid cross. Now, one more MCQ I will tell and I will close the class. Now, Another MCQ, we can learn types of genotypes. Simply genotype number will write. Genotype number in F2 of dihybrid. Or if a question was confusingly asked as types of genotypes in F2 of dihybrid, then also the answer is, we have got just before, if you go back and see the video, 1:2, 2:1:2, 2:4:2, 2:2, again 1:2:1. This one you can learn in your by framing your own small logics. Okay, so how many types? 1, 2, 3, 4. Yes, I'm not counting the numbers, I'm telling the types. 1, 2, 3, 4, 5, 6, 7, 8, 9. So genotypically, what is the number? 9 types of different arrangement of alleles. This one you can learn as an MCQ. Okay, so here I have finished up to the dihybrid cross of Mendel by taking Mendel's third law, law of independent assortment example, and I have explained. I think I have covered all the MCQ models also. Even the new students who are learning genetics for the first time will easily understand this type of explanation. We will see about trihybrid cross and also different types of crosses like test cross, back cross, reciprocal cross types of crosses in the next video.